Hello, everyone. My name is Alex Pueschel, and today I'll be presenting on elucidating tumor cell interactions with Cy5 in microfluidic tumor models using fluorescence microscopy. So about the tumor microenvironment, it has the characteristic of enhanced permeability and retention effect. On the left is healthy vasculature. On the right is tumoral vasculature. You can see it's windy and all over the place. It leaks as indicated by these dots, which are macromolecules that have leaked out. And they stay there due to impaired lymphatic drainage. So if they leak out, they probably won't leave. So then there's the current testing strategy to go from 2D cell models to inform animal models. However, 2D cell models don't very well inform that, and that's because the cells are grown in a flat environment and they don't incorporate a lot of biological characteristics such as dynamic flow. These are under static flow. However, they are cheap, reproducible, and easily imaged. Animal models, however, they have variability between the subjects. The animals are under anesthesia, which has ethical concerns, and they can probe organ-level interactions. But we suggest going from 2D cell models to 3D microfluidic tumor models to better inform animal models. Now, these MTMs allow cells to grow in a 3D environment. They mimic the enhanced permeability effect, something 2D models don't. And they can have co-culture, which is you can have more than one cell type grown in this tumor chamber, or one in the tumor chamber, one in the vascular chamber. And the tumor chamber is in red and the vasculature is in blue. How do we do this? Well, first we degas the MTMs by filling them with a saline solution and then using a multi-port manifold and pneumatic primer to flow nitrogen gas in. And that increased pressure helps force the air bubbles out. Next, we seed with a matrigel and seed coating, where cells are mixed with the matrigel and perfused or injected slowly. And then that helps create a 3D environment and a basement membrane for the cells to grow. Next, we fix the cells and image them. Fixing adds a fixative agent, which freezes them in space and time, and the system no longer changes with time. And then we use a fluorescent microscope. Here you can see the tumor chamber with cells fluorescently labeled in green. And on the right, we have the tumor chamber again. Again, you can see the cells, but the matrigel is now magenta because the Cy5 that we introduced to probe cellular interactions only stayed extracellularly in the matrigel and did not get taken up by the cells. Here, we proved that the cells formed a 3D culture by taking an XY cut and a YZ cut and an XZ cut, which shows the 3D structure using two-dimensional planes. And then next, we have a heat map where warmer colors indicate height off the bottom and cooler colors indicate closeness to the bottom. Again, this fluorescence analysis study was done. This time we turned the magenta labeled matrigel into this white surface. And as you can see, the cells are now cyan and magenta spheres. The magenta spheres are ones that are touching the white surface, and cyan are the ones not touching the white surface. What do we conclude from this? Well, multi-inlet degassing gives air bubble-free chips every time. The syringe pump allows for good control. Doing it by hand introduces a lot of error. There's minimal cellular uptake of the Cy5. And as you can see, we've also confirmed and achieved 3D culture. And this is proof of concept showing that we are able to grow cells and image these. What do we want to do moving forward? Well, we want to further develop our protocols by improving the cell fixing technique, but even further, we want to build on that to take live imaging so we can see how the system changes with time, even up to 24 hours. We want to effectively culture endothelial cells and even seed them in this environment in the vascular chamber in the blue. And that will give us a more better representation of the EPR effect. Next, we'd like to increase the amount of time for uptake of Cy5, which will allow us to see if more time was needed to allow for uptake. And then we want to standardize the protocols so that we get the same results every time, and also they help us enhance the protocols. Finally, we want to put these into practice. We want to use them to test our in-house MNO nanoparticles to see if the liposome encapsulation or the PLGA has an edge over the other and what other characteristics they have. Also, we want to have viability and cytotoxicity to see if the nanoparticles are killing cells. And finally, we want to see the tumoral interactions. How is it interacting with itself and other cells? Thank you. Please, if you have any comments or questions, leave them below.